These are bioindicators for our exposed zone, as they naturally collect a variety of neighborhood identifiers that both bees and humans come into contact with in the 2-mile radius of their hive, thus creating a dynamic map of their ecosystem that would otherwise be impossible for humans to create. I started this project in 10th grade when I was curious if the DNA found in honey could be identified. I worked with Coulter Harbor Laboratory to extract and sequence the DNA from samples around New York City. After that, I wanted to expand my samples to more diverse ecosystems and collect samples from around the world. Bees collect a diverse group of microorganisms from their surrounding ecosystem that impact their hive when they return from pollinating. These microorganisms are incorporated into the hive, which may in return serve as a biomarker for bees' health, as the microorganisms they come in contact with can be identified through DNA sequencing. Bees are dying at an alarming rate, so it's important to identify the contaminants they may be encountering and develop a new way to explore ecosystem health. I capitalized on the partnership between my high school and NYU to improve the depth of my research that greatly increased my engagement and performance in STEM. There were 2 parts to this project over the course of 3 years, and 37 samples were collected internationally. We organized them into 3 groups based on regulations for pesticide use, the organic labeling criteria, and their type of ecosystem. The red group refers to locations that most likely have pollution. Green is least likely to contain pollutants, and yellow is commercial honey that has a variety of levels of regulations. Specific details for grouping criteria are outlined in Table 1 in the center of the poster. In our results, we show a subset of the bacteria and microorganisms found that are known to be indicators of bee health and pesticide use. For example, lactobacillus, which has been shown to increase in the presence of pesticides, was found in a higher concentration in the red group samples compared to the other samples. Enterobacteriales and Burkholdeриales were also found to increase in the presence of pesticide used to attack varroa mites. Our data shows that there were higher concentrations in the yellow and green groups, possibly indicating an increased population of varroa mites in these areas. On the outset of study 2, we predicted bacteria with a connection to pesticides to be present in our samples, or samples collected from the red zone with greater amounts compared to the other groups, especially the green group, and in greater amounts compared to beneficial bacteria. However, at this point in our analysis, the yield from our DNA sequencing provided an array of bacteria that we must first explore in its entirety before commencing statistical analysis to explore significant differences in our specific research hypothesis. This project aimed to not only compare honey samples across ecosystems, but to also learn analytical strategies to exploring whether bees' honey health can be tracked through the DNA of their microbiome, in this case, honey. Bees collect more indicators of their environment than would ever be possible for humans to manually find and collect. This information is compiled in honey and turns it into a biomarker to understand what we are exposed to in our close surroundings on a daily basis. Furthermore, in collecting and sequencing these samples, we will create an open source database for use by researchers in the field to compile and refer to. Looking forward, this model can be expanded in many different ways, including investigating the honey for chemical contamination or looking at the RNA. Using bees as a biomarker could have much larger implications for understanding human health in particular ecosystems. Finally, this project may be a viable model for other high schools with students interested in STEM, especially for future scientists who may be underrepresented in the field, to build relationships and mentorship opportunities in the research community. Thank you, and if you'd like to learn more or get in contact, please scan the QR code and it will lead you to my website with my references.